early today, and so we won't take a long break in the middle, but we'll have a nice stimulating discussion on allergy. I brought a course of allergens to the class. Hopefully everybody survives okay. She should settle down, but just stop petting her if she's bothering you.
Never.
So next week, after next week's lecture, we'll be halfway through the class. And next week is the end of the nuts and bolts, the mechanics of immunology. So everything after that will be fun. Disease and applications of what we've learned so far. So we will have gotten through all of undergraduate and even graduate level immunology in the last 6 weeks. I know a lot of it won't have stuck yet, but we'll keep applying it. So today we'll go over the quiz. In general, you guys did pretty well. There was one bad question, which everybody got right, which I'll point out. There are a few questions from last time that I will try to answer. And then we'll spend the rest of the day talking about T-cell immune responses. And there's a lot of, in the first part, we'll talk about rearrangement of the genes of the T-cell receptor. And that's going to be very much like the B-cell receptor story. And then MHC, which is sort of nothing like the B-cell story. And we'll have the discussion in between. So feel free to interrupt or step out if you need anything. Okay, so question 1 on the quiz. Membrane-bound pattern recognition receptors can be localized in all of these locations. The TLRs are on the membrane and the endosome. There's a lot of cytoplasmic ones as well. These are all pattern recognition receptors. So all of the above is the answer. Ian.
Yeah. Oh.
Hi. You're in big trouble.
Yeah.
So the second one, TLRs only respond to pattern recognition receptors only respond to non-self. That's not true. There's a lot of self molecules called DAMPs that they respond to. So most of you guys got those two right. The third question was a bad question. I was trying to point out that T cells are only specific for protein antigens. They do only respond to peptide antigens, but they can't respond to peptide antigens in the absence of MHC. So true or false were both correct answers. Don't be afraid to write an explanation for your answer on the test. And then number 4, B cells require antigen presentation. In fact, this was a slide and a figure from the book. So it does not require antigen presentation. The antibodies and the B cell receptor can directly interact with antigens without antigen presentation. And then the antigenic specificity is determined not by the light chain, not by the heavy chain, but by the variable region combination of the two chains. Any questions about the quiz?
When you say antigen presentation, does that imply like an MHC is presenting it or some other helping thing?
Another cell, yep. Anybody else? Okay, so I sent out the test on Wednesday. Should be self-explanatory. Are there any questions about it? Feel free to email me. I won't give you, there is no right answer. There are well-supported answers. Hopefully you can support an answer in a long paragraph. Doesn't have to be long if it's very dense. Please email them to me by before class next week, and then you'll go on break. You are allowed to stop.
I can't though.
And feel free to email me any questions you have, although the more notice I have, the easier it is to answer your question. think that's about it. Cody, this is the answer to your question about what the
How's that?
dots and lines. I didn't understand his answer, so I just copied it. It's basically about the potential molecular diversity that can be encoded. You can read it and explain it to us later. It's about how much variability you need to cover all the potential variable space, and that was the overarching theme of the BCR recombination, was how much diversity was required. We had to have a lot of different genes, we had to combine them in different ways, and we had to have a lot of mutations involved in the process to cover potential molecular space. Right. And then the question was about whether this is a single or a double-stranded process. It appears to be a single-stranded process, so there's actually a melting event, so that there's base pairing to make these stem loops within the same chain. Okay, anything else before we get started on new stuff? Relatively new stuff. So this is a little bit of review of T-cell, which we'll finish next week, and go on to applications. Okay, so most of what you learned in the B-cell lectures can be applied, with a few exceptions that I'll try to point out. The structure of the T-cell receptor is very similar to an antibody or the B-cell receptor. There are multiple chains. There's a constant region, and there's a variable region. One important difference is that there's just one binding site on the T-cell receptor. So we know that the different antibodies, the basic monomer of an antibody has two binding sites, and then there are isotypes like IgA and IgM that have even more binding sites. There's just one on a T-cell receptor, and there's no soluble version. The B-cell has secreted antibody and cell-associated B-cell receptor, whereas the T-cell is only cell-associated. Otherwise, their domain structure is similar, and the organization of constant and variable regions is very similar. So just like we talked about for antibodies, the complementarity determining regions, these variable loops at the tip of the T-cell receptor is what determines the molecular shape. Although there's not nearly as much variety as the antibody, because it only has to interact with MHC and peptide. Okay, and the gene organization is very similar to B-cell as well. We talked about how there's sequential organization of the light chains and the heavy chains. And in the light chains of an antibody, you have to pair a variable region, of which there are many options, to a joining region, of which there are many options. Once that combination's taken place, then you can transcribe and splice RNA. In this case, however, there's just one common region, as opposed to the various isotypes of antibodies. So that's the alpha chain is one V and one J, and the beta chain is a V, a D, and a J, just like the heavy chain of the antibody, suggesting perhaps this was a single receptor that then diverged evolutionarily into the T-cell receptor and the B-cell receptor. So you don't need to know how many there are, but just know that the beta chain is like the heavy chain, and the alpha chain is like the light chain. So the result of those combinations, just like in the B cell, where you have a random combination on the light chain and a random combination on the heavy chain, you have those two combinations in the alpha and the beta, and then those can be paired in different ways. And so that's exactly the same way diversity is generated in T cells as in B cells, by splicing and recombining the DNA on the chromosome. So just like for B cells, every daughter of a mature T cell expresses the same T cell receptor. And just like the B cells, it's the same RAG-dependent molecular steps involved. The combination of the RSS sequences to loop out the intervening DNA, excuse me, splice it out, and then rejoin the chromosome in the region of the T cell receptor. So exactly the same process, same enzymes, the only difference is the number of binding sites in the variable region and the fact that it's only cell associated and only associated with T cells. This is redundant. There's the same pairing of the 23 base pair loops and the 12 base pair loops to signal where the variable and joining a diversity region should be looped together in order. And again, the same enzymes do the process. We've seen this a number of times now. So any questions about, so that's much faster than we went through it for the B cell, but everything is exactly the same, except it's a different chromosome, a different set of genes, resulting in a receptor for different cells. Kai?
I really heard learning about the isotopes of the heavy or constellation. So are there any isotopes or
So there's just a single alpha and beta constant region. There's not different isotypes like the antibody, because there's really only one effector function, the signaling through the cell membrane, whereas because secreted antibodies have different effector functions when they're circulating in the body, and the T cell receptor only has functions associated with the T cell. Ian?
Step back a little bit, but for the different isotypes of the antibodies, does each individual one kind of have its own gene pool of things that are recombined to make them, or are they, or how? What's the difference, or like?
So in every cell in our body, including B cells, there's one. Well, there's two chromosomes, right? So chromosomes in pairs, and each chromosome has all the constant regions on it. So when you combine the variable region, you still have all those constant regions. And so then by splicing, you can make different isotypes from the same variable region. However, once the B cell has class switched into the isotypes that are farther down the chromosome, it cuts out some of the constant regions, so then it can't go back. But there's just the 5 choices, the delta, gamma, mu, alpha, and epsilon. So those 5 isotypes. The immature B cells only make D and M, then when they class switch to G, A, E, they can't go back to D and M.
Oh, so this is the isotype relates to the constant region, or
Exactly.
which constant region it has.
Exactly. Okay? Good. So just like the B cell receptor, so when an antibody is in its membrane-bound form, it's called the B cell receptor, and there are other chains associated with it that allow it to signal into the cell. The alpha and beta chains themselves don't have a long intracytoplasmic region. They don't have a region that can actually signal. So they use these other chains to form a complete signaling T cell receptor, so that when this variable region binds its antigen, it needs all of these accessory proteins to tell the T cell that it's seen its antigen. So a functional signaling monomer on the cell involves one delta chain, one gamma chain, 2 epsilon chains, and 2 zeta chains. And these extracellular parts form what's called CD3, which is a cell surface marker exclusive to T cells. So you can identify T cells in blood or other parts of the body by scanning for antibodies against CD3. So only T cells have the T cell receptor and CD3. Okay, so there are actually 2 types of T cell receptors. Alpha beta is the typical one that we've been talking about. Gamma delta is less commonly used. It's in some subsets of T cells that we may or may not talk about later in the semester. Not important to worry about at the moment, but it seems to have arisen from a duplication of the entire TCR region of the chromosome. So inside of chromosome 14, where the alpha and beta, where the alpha chain is transcribed, the gamma delta regions are inserted into that locus. So all of the gamma delta variable joining and constant regions are inside of the alpha chain locus. And the gamma chain is on an entirely different chromosome. So nothing different in terms of the mechanisms. It's just T cells have either one or the other. They have alpha beta or gamma delta. Most are alpha beta. Otherwise, everything is the same. So it would be as if there were totally different but related types of B cells, but there aren't. It's a duplication of the entire TCR region within the chromosome. Okay, so just like we talked about for the B cells, adding the random recombination of all those variable regions, all those diversity regions, and the constant regions is how you generate the kind of diversity that you need. So in antibodies, all that math ends up with a potential combination of 2 million variable regions that then can be joined in different ways to increase the diversity, and then mutated even further to increase the diversity. Same with the T cell. You have about 3 times as many potential gene recombinations. You can join them in even more different ways, and then you can get mutation to add further diversity. So the potential theoretical diversity of the T cell receptor is even higher than the B cell, although the numbers are kind of, I can't imagine what something to the 13th, what that number means, but they're huge potential opportunities for variation. Okay, so that's pretty much it on the generation of T cell diversity. We went through it much more briefly with the B cell diversity, but most of the same rules apply. Let me know if you have any questions. The rest of today will be about the interaction of, because T cell receptors can't operate extracellularly, the rest is going to be about how they interact with peptide associated with MHC. Okay, so the TCR only interacts with protein antigens, and it only interacts with protein antigens when they've been processed into short peptide sequences in the order of 10 to 15 amino acids and expressed on a molecule called MHC. So cells that present antigen to T cells break up proteins either from an extracellular or intracellular source and have to get them out on the surface to show them to T cells so the T cells can interact with those peptides. So T cell mediated immunity only occurs in reference to other cells. So we mentioned last time, and I'll mention again, that CD8 positive T cells interact with class 1, which presents the self or intracellular peptides. So CD8 cells are moving around the body looking at peptides associated with intracellular pathway to see, is this self cell making something that I don't recognize, like virus, so that then it can form a direct reaction against the virus-infected cell and kill the virus-infected cell. CD4 cells typically only interact with professional antigen-presenting cells, so-called macrophages or dendritic cells. Those are the ones that are able to take up extracellular proteins and pathogens and present them on class 2. So intrinsic CD8 class 1, extrinsic CD4 class 2. And then the response of that T cell, when it's seen its antigen for CD4, it can make cytokines to enhance the macrophage's ability to kill and make its own cytokines, or it can make cytokines to promote B cell maturation. And we talked about how a full T-dependent antigen requires 3 signals to form a B cell response, and one of those signals is the presentation of antigen to T cells and cytokine help in order to inform. So the T cell typically doesn't act on its own, it acts in reference to other cells. It kills cells that express non-self proteins on class 1. It promotes inflammation when antigen-presenting cells show it for antigen, and it interacts with B cells to form a complete immune response to foreign, you can mean antibody response to foreign molecules. Go ahead, Jen.
Sorry, just are CD4s all called helper T cells, or are they all called helper T? CD8's not called helper T.
CD4 cells are helper T cells. CD8 cells are cytotoxic T cells.
Okay.
Good point. Anybody else? Ian?
So when like a other cell presents something, or presents MHC with a little segment of foreign DNA, does that
Foreign protein.
Sorry, yes.
Yeah, okay.
What was I saying?
Yeah.
Does that mean that there are like restriction enzymes, like kind of like bacteria have? They're in cells and like they have their own ability to recognize foreign material?
Yes and no. So we're going to, as we go through some this lecture and some next lecture, we're going to talk about the enzymes that break up proteins. They aren't sequence specific like restriction enzymes are. They're more general to sequences that are repeated in lots of proteins. So they're proteases that tend to be able to digest most proteins. So they're not quite so specific to an exact amino acid sequence. But
So-
yes, that's a reasonable analogy.
So we have a lot of MHC cells that are just randomly presenting protein segments that could just be us.
So, right, so let's handle the class 1 and class 2 separately. Class 1, pretty much every cell in your body, with a few exceptions, has class 1. And some portion of the protein that it's making at any given time, it digests and displays on the surface of class 1. So it's basically always showing off whatever proteins it's making, because it doesn't know what's self and non-self. A cell in your lung, a cell in your lung or a cell in your gut doesn't have the immune system's ability to differentiate between self and non-self. So its job is just to show a small amount of whatever it's making at any given time, usually itself. But, and the CD8 positive T cells pass by and say self, no response, because they don't have TCR specific for self proteins. In the rare occasion when that cell has been taken over by virus, it's making some proteins that are not self. And so while it's throwing bits of those onto the surface, then the CD8 cell comes along and recognizes it's not non-self. So some viruses actually have mechanisms to reduce class 1 expression. So basically when I infect you, you stop displaying peptides on class 1, so it can't be eliminated. So that's the class 1 story. Class 2 is only on dendritic cells and macrophages, and they really only go through that process when they've engulfed something foreign. So they get a bunch of signals. I see there's a bacteria, it's activated my TLRs, I do phagocytosis, I kill it, I digest it, and then I go through the process of putting stuff out. So that's a more of a on-demand system, whereas the class 1 is a constant. Okay. So, right, so we've got the T cell receptors on CD8 and CD4 positive T cells. And then, as we've been discussing, there are 2 classes of MHC, class 1 and class 2. They're similarly structured. They have alpha and beta chains. Class 2 has 2 transmembrane regions, class 1 only has 1. Doesn't really matter. The point is that these are not these are not specific to a peptide. Lots of different MHC molecules can present lots of different peptides. So unlike antibodies and T-cell receptors, which are exquisitely specific, you can load the short peptides into many different class 1 or class 2 molecules. As you can imagine, we can't know in advance every protein that exists in nature, so these have to be fairly flexible to be able to present different kinds of peptides. Okay. So this is just a review of what we were just talking about. The place that CD8 and CD4 not only mark the cytotoxic and the T helper cells, but they're the molecules that directly interact with the constant part of MHC. So there's a whole bunch of complex interactions. So when CD8 cells interact with self cells expressing antigen, part of that molecular interaction is the binding of CD8 to MHC, as well as the variable region interacting with the peptide in the peptide binding pocket. Likewise, CD4 helps bridge the gap between the helper cells and the antigen presenting cells, whereas the variable portion is interacting with the peptide. So this is a highly variable interaction. This is a constant interaction. Every class 2 molecule looks the same down here. Every CD4 interaction looks the same up here. And the specificity comes from this variable interaction. Okay. So this basically gets to your question, Ian. So in the cell, all the time, we're translating protein, and most of that protein gets kept in its intact form and goes on to do whatever its job is. But some small percentage of it is digested by the proteasome, which has a lot of responsibilities for breaking down proteins, and but it also has an important immune function in that it digests all the proteins we're making into peptides, and then a membrane transporter called TAP transports those proteins into the endoplasmic reticulum. So from the cytosol into the endoplasmic reticulum, and that's the site where they can get loaded onto class 1. So this will come up again in the next class, and this is the sort of thing it's good to have a reference textbook. You would, unless this is what you did your research on, you'd never have to draw this series of steps, but not surprisingly, it's a highly regulated process in that class 1 gets expressed inside the endoplasmic reticulum. It's stabilized by a bunch of accessory proteins, and then once it gets loaded with peptides coming in from the proteasome, those accessory proteins are released, and one of the functions of those accessory proteins is to keep class 1 in the endoplasmic reticulum until it has a peptide loaded on it. And then once it has a peptide loaded, it can be released from the chaperone proteins and then moved to the surface of the cell. So the construction of the complex occurs in the endoplasmic reticulum, and then it's trafficked on a vesicle to the cell membrane. Okay. And sometimes those peptides don't fit perfectly into MHC class 1, so there's actually some more proteases that can trim them down so they fit to be the right size on the, so basically you could load a longer peptide that gets trimmed down to the right size for the MHC before it gets moved to the surface. Okay. So everything we just talked about with class 1 happens on the right side. Intracellular antigens, digestion by the proteasome, pumping into the ER, loading on class 1, and then movement to the surface. Class 2, we don't have to load the proteins into the endoplasmic reticulum because they're already there. We basically took up an extracellular antigen into a membrane-bound vesicle. That becomes a phagolysosome, so we have an oxidative burst and the release of a bunch of proteases to break up the extracellular pathogen. Then, again, in the endoplasmic reticulum of antigen-presenting cells, those get loaded. So essentially, it's a simpler process because those foreign molecules are never in the cytosol. You guys are really quiet today. Does it make sense, or is it too much to even ask the question about? Kai?
In previous slide, when it's like cleaved,
Yeah.
will the other ones be recycled, or will they like be excreted?
The amino acids that get cleaved off, I assume that they would be recycled, but I don't know this process to that level of detail. It's unlikely, it takes a lot of energy to synthesize amino acids, and you can't even make all amino acids, so I would assume they would be recycled. Ian?
Is there, could you go back to the other slide? So does MHC class 2 vesicle like the combination of like ER and like lysosome or something? Just so you can get that like, you know, breaking
Right.
up of the foreign
So,
antigen?
if you remember from some of the innate lectures, when the cell does phagocytosis, it brings in that foreign extracellular antigen in a phagosome, and nothing's happened to it yet until it fuses with the lysosome. The lysosome brings in all those oxidative burst enzymes and proteases to kill intracellularly. So that's when that vesicle basically brings the class 2 in, along with the other molecules that kill the pathogen. Angelique?
If you go back to slide 25, I noticed that the two types of MHC, like class 1, class 2, like one of their differences is like their subunits. And I was wondering if the difference between their subunits will allow one to have a higher affinity for a particular antigen than the other. And if not, how is it that they can have different subunits and then still interact with the same antigen, like equally, I guess?
So that's a really good question. So, just like the TCR and the BCR, the constant regions, these ones closest to the cell, are not variable. So they have every MHC molecule has a very similar interaction with CD4 or CD8. This end, we'll find out in the next lecture, we have hundreds and hundreds of genes for MHC. We have lots of different genes. So there will be peptide MHC combinations that are more stable and ones that are less stable. So some peptides will get loaded into some MHC better than others. And then the T cell receptor, its specificity is for that combination of MHC and peptide. So some of those combinations will have a higher affinity interaction than others as well.
So does one class have a higher affinity than the other, or?
I don't
is it
think
really
so.
not? Okay.
I don't know that off the top of my head, but I don't think so. Jenny?
So once the MHC interacts with the T cell receptor, does T cell then go through like a clonal selection, kind of like the B cell, or does
Exactly.
it?
So
Okay.
each T cell just has one T cell receptor, and then you activate and proliferate, and you have somatic hypermutation and all that other stuff that happens in a clonal B cell.
Isn't it sort of inefficient to have different receptors for like different peptides? Would it be more efficient to just have like a broader type receptor to just accept every peptide and just be like, this is not me, I'm going to the B cell?
Would be more efficient. Why don't you think that's what we do? Kai?
Because we need specificity to attach to different types of peptides.
It could be, that gives more flexibility, but if we all had only one receptor, one MHC, then the pathogen it would be pretty easy to evolve some mechanism to defeat that, right? So basically, there's a section in this chapter about the evolutionary pressure that infectious diseases probably place on the system to result in so much gene duplication, because every time we had some way to identify a pathogen, he had figured out some way to defeat that, so we had to have sort of infinite variability. Okay, so similarly to the to class 1 loading, the class 2 has a protein, it's actually a membrane-bound protein called the invariant chain, and it basically occupies the MHC, stabilizes it until it becomes part of the phagolysosome. So those enzymes that digest foreign pathogens also digest the invariant chain, such that the foreign peptides can displace the invariant chain on its way to the surface. This is really a whole lot of hand waving. I mean, this is what they see happen, but exactly how it's regulated is not entirely clear. It's clear that both class 1 and class 2 have to be stabilized by other proteins until they're loaded with peptide. And at some part during this process, this part is, the invariant chain is digested so that foreign protein can get loaded onto it. And there's other accessory proteins that help stabilize it as well. But basically, it's a protein that is only really stable when there's a peptide in the binding pocket. So there's various mechanisms to stabilize it until it gets loaded with peptide. Okay. So then we've been talking about how class 1 is expressed by most cell types. So this includes the immune system, so lots of hematopoietic cells express class 1, and most other tissues express low levels of class 1 as well, except in the case of an infection or inflammation when they upregulate their class 1. So if you get a viral infection, you have a cold, the cells of your airway will increase their class 1 expression in order to try to increase the likelihood that those viral proteins get presented. Class 2 is restricted to cells that can be professional APCs, B cells, macrophages, and dendritic cells. And then, of course, thymic epithelium is a unique non-hematopoietic place that has class 2. Why would that be important? Ian?
Creating our cells that recognize self.
Right, exactly.
Or
that's
rather,
where,
yeah, immune system.
that's why we're doing central T cell tolerance. So there's lots of antigen presentation there. So if we didn't have class 2 in the thymic epithelium, we wouldn't be able to educate CD4 positive T helper cells. Kai,
There is no T cell at those sites.
can't hear you.
So why isn't there any presenting class 1 and class 2 in erythrocytes?
In red blood cells?
Yeah, blood cells.
Well, because their job is pretty much to, they're non-nucleated cells. Their job is to transport oxygen. That's how they evolved. I don't know what the best answer to why is, but they don't. So when you get a blood transfusion, you can be MHC mismatched to the person you get red blood cells from and not have an immune response to them. So this is a whole different system than the blood type antigens, A, B, and O, which are expressed on red blood cells. Okay, so that's the first lecture. Take a bio break if you need, and the discussion group will get ready.
Wouldn't it be kind of hard for blood cells without anything to do transcription or hard for them anyways to make MHC? I mean, maybe except for like once they were processed fully and like the nucleus was taken out.
Yep, that's true.
Is there a disease where something infects the thymus and then your immune system is compromised because your cells develop tolerance to forest particles? Probably because
Intrinsic and extrinsic
Dude, I don't know how to do, I didn't know how to do this.
I couldn't figure it out. Because I thought like
I just
there's
couldn't figure out
blood
how to make it
cancer,
this
right?
long, so I made up
Like
something.
blood cancer, right? So is that the blood, like which
Like, I knew
part of
everything,
blood is
like
Depends.
from
Could be
here,
T
like, I
cells,
know how
could be
to
B cells, could be
here,
monocytes.
so I
That's
just
why
didn't
there
know
are
how to
different
from here.
names for leukemia.
individual.
If
Yeah,
it's a T
two
or a
carbon
B cell,
blood,
it's a
just like
lymphoma.
put it in the
If it's a myeloid leukemia, it's a neutrophil or a monocyte.
I don't know how.
So
I don't know if I'm.
it depends on what white blood cell is proliferating. We'll have a whole class on immune cell cancers.
Okay, so red blood cell can also have a cancer?
No, because they don't have any DNA. They don't
So
have
that's why.
nucleus.
Okay, I see. Yeah, okay. That was the question.
Okay, so I guess my question, I mean, I was trying to ask last night, but is there something about the nature, since, I mean, you know, there's kind of a common pool of, like, allergies we have, like, very specific things that you run some allergies. Is there something about the proteins that allergies are developed to that give rise to the IgE? I mean, like, so is the gene or the specific parts of our genome that are used for IgE variable regions, is that different than like IgA?
It's not about it's about the kind of immune
A lot closer than it could be.
It's about the cytokines that are made in the T cell that causes class switch to IgE.
So wait, what?
It's the kind of cytokines that are made in the immune response that causes class switch into IgE.
What causes the cytokines to be made?
Or some of the types of antigen is part of it.
Got
The
there.
route of delivery, the types of antigen,
Are you taking Orgo
the
now?
right time to make those kinds of reactions more than somebody else.
And so, does that mean like you might develop allergies to, or not allergies, but like
I think if I took Orgo now, I'd be more interested in
like
it.
pathogen, but it
But
wouldn't really have a noticeable effect.
I like no chemistry background coming here basically.
Just because
So I
like it's not like gonna create IgE. Okay.
Exactly. Yeah, so most,
Yeah,
that's
I
what
never
most
looked
of your
into it.
IgG and your
But I was like-
IgA are taking care of those foreign substances all the time. You don't know the difference.
Just that it's not.
Oh, you stick it past fail like one.
Thank you. That
Yep.
answers my question.
Sorry, just a quick question. When you said that some of the viruses have like class 1 signal dampen, so they don't present anything,
Right.
isn't that just as easy to detect as if they're presenting like normal cells normally? Can't they also tell if there's nothing being presented
Very
at all?
intuitive. There are mechanisms for seeing that there is no class 1. But a lack of class 1 doesn't stimulate the T cell receptor. There's some other mechanism for it. There's no, the T cells don't know, oh, I'm not seeing class 1. I'm seeing a cell, but I'm not seeing class 1.
Okay.
There's other immune cells that, NK cells are good at noticing there's not enough class 1.
Okay. Wait, does it dampen production of class 1 in general?
Yeah.
Okay. I thought you were saying it just dampens the presentation so that there be nothing being presented as opposed to, okay. But even so. Alright. Thank you.
Okay. Take control when you're ready. Okay. Okay.
I think I'm presenting to one.
You guys man.
Oh, I went for like what?
I'm gonna go stay after this.
Why?
Right after.
Crazy.
Don't ask him why, then.
How did you know?
I'm not dark.
So I'm like walking out of the someone's like without power.
What is your dog's name?
Jenny.
Jenny? Jenny.
I think dad like
He's not gonna leave that.
Yeah, I was there.
Actually, I like Oh, yeah, exactly.
Hi, guys.
Their loss.
My group and I, we studied hypersensitivity, and more specifically to the article that we sent out for you guys to read, is more based towards increased prevalence of food-induced allergy. So just as some background information, hypersensitivity is also known as hypersensitivity reaction or an intolerance towards something. It refers to the undesirable actions produced by the normal immune system. But what this requires is a pre-synthesized immune state. So what that means is that you have to be exposed to some type of antigen prior to developing a hypersensitivity to it. So what this leads to is to the production of allergies and autoimmunity, such as autoimmune diseases. And these two repercussions may be damaging, uncomfortable, or even lead to fatal outcomes such as death. So allergies occur when the immune system reacts to normally harmless substances within the environment. So some examples of this is pollen, dust, pet hair or dander, or food. I know you guys know things such as celiac disease, or otherwise gluten-free. People who have allergies in the springtime due to an overpopulation of pollen, and everyday pets such as dogs. So autoimmunity occurs when the body fails to differentiate between self and foreign, and as I aforementioned, celiac disease is an example of autoimmune disease, and that's essentially because your body is attacking its own cells. More specifically, it occurs within gluten-free people, and another example of autoimmune diseases is type 1 diabetes, which is the body's destruction of its own insulin-producing beta cells within the pancreas. And the third example of this is rheumatoid arthritis. It's basically when your body attacks its own joints and breaks down the cartilage. So it leads to your joints fusing or rubbing together. And so fusing is a latter stage. It essentially starts with the rubbing together of your joints due to the breaking down of the cartilage between them. So there's 4 types of hypersensitivity, and Nancy will take over from here.
Okay, there are 4 types. First, type 1 is allergies, and it's known as intermediate, immediate, and basically like it's just like normal allergies that you would have to like, I don't know, like different food and stuff, and it happens immediately, which is why it's type 1.
That's what we'll be talking about, by the way, in a little bit.
Yes. Yeah, so we will go into detail about that. And type 2 is cytotoxic, and this is basically when the antibodies produced by the immune response binds to the antigens on the patient's own cell surfaces. So, for example, like if you take penicillin, sometimes the drug can bind to your red blood cells, and then you can have like a immune response to that. And then type 3 is immune complex, and that is when the antigen-antibody complexes are not adequately cleared by innate immune systems, and it causes an inflammatory response and leukocytes to accumulate. And an example is farmer's lung where you like inhale antigens. So basically like if you inhale like mold or like dust, sometimes you'll have an inflammatory response and yeah. And type 4 is known as cell mediated or delayed type, and that's like a lot different than the other ones because it's cell mediated, and usually it takes 2 to 4 days to develop. And so when you like catch poison ivy, or sometimes when you get like the TB test, you'll like develop some kind of rash a few days later or sometime later, and that's type 4 hypersensitivity.
A major difference of type 4 in comparison to the former 3 mentioned is that obviously the delay time is much longer, and the I guess like the effects of it is more superficial, so you'll see it more on your skin, like as in rashes, rather than towards like swollen glands, even though that can still occur, but it's more, its first presence in which you'll see it is like in the form of rashes or bumps or having some type of itch.
Okay, so as Ian mentioned and alluded to, our presentation is going to focus from here on out primarily on type 1 hypersensitivity because that's what the article's on, so it'll make for a better discussion. So type 1 hypersensitivity is caused by re-exposure to an antigen. So generally the first time you are exposed to some sort of foreign particle, the response isn't too significant, and many of us have friends or have heard that usually like someone, for example, who will eat peanut butter and they'll have some sort of immune response, perhaps hives or itching or just like a rash on the skin, but the next time it'll be worse, you know, and ultimately results in anaphylaxis. That's the most severe, which can result in death. Common allergens include pollen, mold, dust mites, grass, some foods, and then certain medicines. And again, these are immediate reactions, so the reaction itself will take place in within seconds to minutes, so very quick. In terms of developing an allergy, like I said, it's in response to like multiple contacts with the allergen. It happens when the B cells respond to an antigen that is not from a pathogen, so it's not coming from bacterium, such as like pollen. We know it's not a bacteria. It's just a release of the protein that we inhale, for example. And instead, so it's like a protein, and the response produces IgE, immunoglobulin E, rather than immunoglobulin A, D, G, or M.
Yeah, and I guess kind of along like, I mean, many other things that don't give an allergy can kind of cause the body to recognize it as foreign, although it's not really harmful. But if that certain substance doesn't give rise to an IgE isotype, then you don't have a problem as much because you're not. I mean, the other types of antibodies don't give rise to massive like histamine releases as IgE isotype does.
As mentioned before, IgE is a major player in type 1 hypersensitivity, which leads to several allergy diseases such as rhinitis and sinusitis and asthma. IgE consists of monomers consisting of 2 heavy chains and 2 light chains, with epsilon chains containing 4 IgE-like consonant domains. IgE primes the IgE-mediated allergic response by binding Fc receptors on mast cells and basophils, but these Fc receptors can also be found on eosinophils, monocytes, macrophages, and platelets in humans. Atopic individuals can have up to 10 times the level of IgE in their blood. However, this isn't necessarily a requirement to have an allergic reaction. Some individuals with asthma can have less than this in their blood, and it's been found in research that IgE can be produced in the nasal mucosa. IgE can specifically recognize an allergen, which is, as mentioned in the past lecture, it's a protein generally that it finds. So just a quick pathway to your allergies. So when an allergen enters the body, it binds to the IgE antibodies, which are bound to the mast cells and basophils. This causes a cross-linking, which gives a release to antigranulation. This is a release of histamines and leukotrienes, interleukins, which then eventually cause your allergic symptoms. So now we're just going to start to turn our focus away from background information, focus on the quick article that we have posted, which is really a quick synopsis of a larger scientific study. The survey was between 1997 and 2008, where they saw a nearly 3% increase in children with peanut allergies. So this is kind of dangerous because peanut allergies generally lead to an anaphylactic response, which, as mentioned before by Alex, is generally death. Sometimes, so it's scary, especially in schools and things like that, where peanuts are always around. Think about commons. The peanuts there, and it goes onto bread, it goes onto a knife here, there, and the next thing you have someone dead in the corner.
Although, I think it's important to note that it's not, while there's potential with today's medical advances, epinephrine and then antihistamines is a pretty easy, not easy, but fine, effective way to manage this. Like as long as you have your EpiPen on you, or EMS is on their way quickly. Yeah. So while I'll get to this a little later, it's not as big of a deal as some people make it, because while yes you could die, it's very easy to treat. Or it's to treat a reaction. Yeah. It's easy to manage. We have medicine. It's not like you give them a shot and it doesn't work. It's not a big deal.
People don't usually die.
Well, it's a big deal, but you're kind of asked to go to the
becomes a
hospital,
big
but-
deal if you don't know you're allergic. That's the issue. But if you know you're allergic, you usually have antihistamines on hand in case you come into contact with one of these antigens.
Do you guys have nut-free housing or areas of commons, or is there any provision for nut
I think
allergies?
Bates is probably a little more reasonable about it. Massachusetts, for instance, I think 2002, I think they established rules and regulations, and I don't know if it's every school, but I know that probably especially lower level schools had peanut-free zones. And so while it's not a huge societal problem, it's more you're putting a lot of resources into something that probably isn't a big deal as long as the kid's careful.
So I have kids in elementary school, and there's always a nut-free classroom in every grade.
Yeah, so it's
the nut-free table.
so it might be a little bit of overreaction to some, it's kind of an emotional response to something that is serious, but is at the same time a societal level not a huge deal, and perhaps too many resources are allocated to something that
Well,
isn't as big.
let's do the science first, and we can deal with public policy.
So there's not much, conclusive theory on why this allergy is on the rise, but there are some theories, and that's what we'll continue to discuss.
Okay, so the article really talks about, briefly mentioned, clean living, and that's basically now that living in the United States, we have a lot of sanitation around, and we would always get vaccinated, and there'd be antibiotics, and so it's it's very possible that it's putting less stress on the immune system, and that so instead of attacking things that they think are harmful, they'll begin to attack things that are not harmful in the body, such as peanuts, not very harmful. And so just by having all the sanitation, it's kind of messing with our immune system, and with the way that we roast peanuts sometimes, it changes the way that our body will identify the proteins of the peanuts, and so that could affect the adaptive immune system.
So it's certainly a problem, and as sorry. So no, it's since it is an adaptive immune response, it's not transferable through the mother. Well, I guess because IgE cannot go through the placenta, but also because it is your allergy is a learned thing through exposure. So if you're in the fetus with a nice supply of blood, you're not getting IgE, and I guess the proteins are a little bit too big to go through the placenta, you won't get, you have to develop it as a child. So there has been studies into like whether or not there's a genetic link, and apparently there is one, although I guess you could kind of say that, I mean, that might be hard to determine because usually, I mean, with a clean living hypothesis on the rise, or as a plausible explanation, usually the parents and the child live in fairly similar conditions, so just, you know, distinguishing between the clean living hypothesis and, I mean, yeah, and a genetic link might be hard to say, but it should make sense, or it does make sense. So we also know that supporting the clean living hypothesis, that people in, well, and the processing of peanuts, we know that like in countries such as Africa, where many regions, they have like peanuts is like a staple, just because they're cheap and they're very nutritious, so when, like, in conditions of poverty, you know, it's a very good food, and actually, when exposed to peanut butter, like probably like Jiffy or something, I think there was maybe a little bit of a rise, or some people did develop peanut allergies, so that would definitely suggest that what roasting does to the proteins may be what is kind of causing the allergy, or, you know, creating this allergy. So some research has been done into addressing this problem. There's a very effective protocol of like desensitization, like through like orally. So if you take a small amount of peanuts at a time, and as you increase the dosage, eventually you can tolerate enough so that, you know, if you accidentally ingest a few, like, I don't know, like you eat a Snickers bar or something on a mistake, it won't be a problem, and that's been effective, although it's a fairly new treatment. And I think that revolves around kind of like central tolerance, you know, the re-exposure, but also that they've I guess there's been the development of drug methods to do this, you know, not just giving the protein, the peanuts, and this is through apparently basically preventing the IgE from getting to these mast cells, and that, I don't know if that's, I think it downregulates receptors. I'm not sure. It's either a competitive inhibition or downregulation of the receptors. Do you know, Aaron?
Do I know?
Whether or not like the drugs such as Xanax, like whether it's downregulating the receptors on mast cells and whatnot, or if it's just competitively inhibiting it.
It's desensitizing the receptors. And
So
the
that-
article that we read was talking about desensitizing the receptors, not necessarily competitive
So
inhibition.
you talked about a bunch of stuff there. So traditional desensitization involves trying to energize the B cells that are making the IgE. So basically stopping the cycle of B cell activation and IgE release, which eventually leads to less IgE of a particular specificity. There are also drugs that target IgE itself, so you can take an IgE neutralizing antibody, and there are also drugs in clinical development right now to try to eliminate all the B cells that make IgE. So basically kill off just the IgE making B cells.
Okay. And then also, all of these methods of desensitization and whatnot are much more beneficial than just, you know, constantly taking an antihistamine, because I mean that has like other like anticholinergic, like systemic effects, which you know can make you drowsy, cognitive impairment. So it's obviously better to have a more specific approach, and one that also targets the problem, I guess, closer, or you know, farther away from the response. Now,
I
next.
just want to say one thing. There's been a similarity between all these articles that we read on this additional research, and is that it deals with, one, if the person has been exposed, and two, the level of exposure. So there's actually a healthy range of IgE that really doesn't really cause hypersensitivity to antigens. And that's actually what one of the things that this article touches upon, and that will lead us to some of our discussion questions. Oh crap, no, sorry.
So Plumpy Nut is an example. It's, for anyone who's not familiar with it or has never heard of it, it's basically like a peanut-based like cereal bar that's given to like underdeveloped or impoverished areas in Africa specifically, so that starving children or malnourished children can have some like source of calories, because peanuts are a great source of calories, protein and fat. And so that also brings us to Ian's point, which he mentioned earlier, that in these underdeveloped countries, it seems that allergies such as peanut allergies are less frequent than developed countries such as America or the UK. And it's not entirely known why that is. It could be genetic, as we mentioned, coming that they're from primarily like Africa, like it's seen well. It could also be just like, as again Ian mentioned, is a staple crop in some regions of Africa. So it could be that they're desensitized to it at a young age. But the quote I put up there, it's food allergies seem far less common in poor countries and rich countries. That's by the creator of Plumpy Nut. And then Dr. Shepard, who's a doctor from Doctors Without Borders, says that he's in his time working over, he's never come across a child who's seen who's had a peanut allergy. And so it's just interesting, and that's not entirely known why yet, but something to think about.
So maybe we could just skip this, but it's more just like kind of like a sociological debate. If you think that's,
we can come back there if we have time about that. I think if you could die from a peanut reaction, that's a big deal. And I think for the purposes of this discussion, there is a real increase in the Western population in peanut nut allergies. How big a deal that is an opinion. But I think if we could focus on why the group or you guys think that might be and what we should do about it, that would be good.
These discussion questions. Alright, so should we take so many precautions?
yeah, just skip that one.
Bye, Ian.
Anyhow, yeah, the second question is on the scientific line. Would it be better to, or go ahead, Dan.
Would it be better to come up with an effective cure or work on prevention, or both?
What do you guys think?
I have a question first. Have you looked at the peanut proteins? Like, do you know about any of them that are cause allergic reactions?
Like us
Yeah,
personally?
did you guys look into that?
No.
We have not.
It's all linked to your development of your IgE. So like, basically if you develop an IgE towards the proteins within peanuts, you're gonna experience some type of anaphylactic reaction or absence.
Run with that, Sarah.
I think there are some different, like there are different proteins that do different, like different like peanut allergy proteins.
Right.
I don't know a ton about them, but I
Right.
think that they
Just
have
because you're allergic to peanuts
reactions.
doesn't mean you're allergic to all nuts.
Yeah, I've heard.
But there's a higher chance of being allergic to other nuts if you're allergic to like peanuts. Like
No, I'm just wondering if you know about like the action with the specific peanut. I mean,
Like how it
it's fine,
works.
but yeah.
What specific protein finds?
Right.
No, we haven't looked
Because
at
There's,
it
like,
would have to be a
I
specificity.
mean,
Yeah.
it
So-
undergoes a type 1 hypersensitivity reaction, if that's what you're asking. That's what the category would fall under.
No, but I think what you're saying is you're not making IG against the entire peanut.
No,
It's
yeah,
some
there's a
specific component.
sequence of
Yeah.
Just
proteins
like with house dust mites, it's not everything in the dust mite, it's a couple of specific proteins.
Yeah.
So
And as someone who has a severe anaphylactic reaction or systematic reaction to it, are they having a reaction to like specific proteins that someone else might be having a reaction to other proteins? Like, is it?
We haven't looked at deep into it, but possibly,
Yeah.
very well possibility, especially if the receptor is interacting with a particular protein. And even though you and I may have an allergy to peanuts, I may be reacting to a different protein than you may be. I mean, maybe.
So let's say everybody with peanut allergy reacted to one protein in peanuts. What would you do about it?
You have to figure out a way
Call
to
it
get rid of
Monsanto.
Genetic engineering, get rid of that protein.
Yeah.
Okay.
I think I might be a horrible person for saying this, but I think working on prevention and giving cures is like actually increasing the allergy. Don't you think so? I mean, like I have an allergy to aspirin, so if I were born in a time or in a time or in a country where there's no aspirin, then I will have like higher chance to die, right? Because I cannot tolerate aspirin. And if there's no any other medicine, then I will, there's a higher chance for me to die, right? So, like, survival of the fittest. So,
Well,
if you, I know, I'm a horrible person for saying
No,
this.
that's actually a
No, no,
great point because the article touched
Enforced
upon
natural
that.
selection?
So, I mean, if there are so many algae products that says, oh, this tastes like peanut, but it's not. And for example, I'm allergic to
Yeah, why
aspirin.
do those exist?
Yeah, well,
So because of that, that's the point that I'm making. Because they exist.
But people need to do research to know that they like have to prevent.
Yeah,
Yeah,
like prevention is
prevention can be-
important, but this is just, I don't believe in this point, but I think this is one of the reasons why there is an increase in allergy.
well, I think that connecting to actually the previous presentation where, like overuse of antibiotics, which kind of to prevent like some of the issues that come along with overuse, you just use them less unless it's like a serious infection. So kind of here, if we actually did that, we would be living, I guess, less cleanly, and our immune system would have more, have to fight more infections for us, and that could, according to the clean living hypothesis, give rise to less of a prevalence. So that would be prevention of sorts, kind of living in like less of a really sterile, taking lots of antibiotics kind of environment.
So
Is that,
I want you guys to get to Rachel's question, but I don't, so you've mentioned the clean living and the hygiene hypothesis. I don't think you've really explained why getting more infections would lead to less allergy, so see if you can do that, but make sure you get to everybody's questions.
Well,
so which one would you do
with the-
first?
I just kind of want to touch upon the level thing quickly. I think it's a check and balancing because if you have too little of something and too much of something, it can both cause negative effects. So I think going back towards the concentration of IG in your body, there's a good range for you to not develop hypersensitivity. And I think that's kind of what you have to think about in terms of everything. The overuse or the use of too little of something can both lead to adverse effects. So I think that's a good way to think about it instead of just saying, oh, let's just take this out completely.
That's the point
then-
I'm making. Like, I'm not saying that we should stop giving alkaline product at all, but I just feel like too much of it
Right.
can be leading to increase in.
I agree with you.
What he's saying is that because we have better medicine,
Yeah.
more people are getting it. Because the people that would have, I don't know how it's if it's into your genes and code or something, but if more people are surviving, then that's
Right.
good.
I
So
think
I think that's what he's trying to say.
Right, I agree with what he's saying. I think it could be argued both ways though. I think someone over here had a
Oh.
question.
It might go in a completely different direction, but have you guys found any research on there's such a high prevalence of peanuts now in our diet? I know that that's a theory for celiac disease. That there's so much gluten in what we eat that people become allergic to it over time, and I don't know if that's the same for peanuts.
I found this cool study.
Uh,
I found a study. Let me say my study because I really want to say the study. I really want to say. Okay, so I don't think that is really the case because in one of the studies they said that Jewish kids in the UK had a higher prevalence of peanut allergy than Jewish kids in Israel. Because in Israel, the Israeli infants consume peanut in higher quantities at an earlier age than infants in the UK who avoided peanuts. And so maybe being introduced to peanuts early could have a good effect. And maybe if we eat more peanuts, we might not develop the allergies. But that's just one study. Right?
Say about celiac, it's a little different because, and it actually almost makes sense that we're having this allergic reaction to it because in more traditional diets, gluten is mostly in grains, and grains are kind of are something that we've basically done through developing agriculture. In the past, we didn't really have gluten, so it makes sense that we're allergic to that, and that our bodies were not evolutionary or have not evolved to consume it.
I'm not sure there's general agreement that celiac disease is an allergy.
There's celiac
Yeah,
and
isn't
there's
it
gluten
genetic?
intolerance.
Yeah.
They're a lot of people are
Celiac
misunderstanding it,
is
and a lot of people are getting way too hyped up about not eating gluten.
So maybe we'll work in celiac disease to another lecture.
So I have a question about the clean living hypothesis. If that's true, would we see a general trend in all allergies? So is this specific to peanuts, or do you see it across all allergens?
We only looked into peanuts. We didn't really look into all allergies.
I think that we're focusing on peanuts, and I guess that is more noted because it's a serious disease. I am under the impression that allergies are on the rise, but it's not maybe as hyped up from a social standpoint because of it's not quite as serious.
Well, it's the same thing for pet dander and all that. It's all rising.
Dust mites, asthma.
Yeah, no, exactly. But I'm saying now we know. That's I guess.
Everything is rising.
In certain parts of the world.
Wait, can I answer the clean living the why, or would you like me to do that?
It's your discussion.
Oh,
You guys
okay.
it's
So
just
I guess
making
the theory behind the
suggestions.
clean living hypothesis, as we had not explained, our bodies, I guess, have evolved to deal with more or less not a set, but some degree of bombardment by pathogens, and with a more clean environment, our bodies have less work, or our immune system has less work. So because it would be kind of bad descriptive word to say bored, but it has less stress on it, so it'll start attacking things that are not really a problem, but because it doesn't have as much work, this will be what happens. I believe that's probably why autoimmunity is also something that is on the rise, or was less of a problem in, I guess, more dirty times.
So I back home I have a number of friends who are allergic to peanut allergies, but they're I don't know if I guess I could say there are different levels. I know one person who cannot tolerate the smell, or if someone has a peanut on the plate, if their plates touch someone else's plate with peanuts, they react. And then there's another person who can't eat it. So I was wondering if the different levels of allergy is due to, I think earlier you guys said that you get hypersensitivity, you're repeatedly exposed to it, or is it because of the different proteins that different people react to when their bodies can't tolerate peanuts?
It's a good question.
So
That's good. Stall, Angelique, stall.
You're asking why some people have reactions that are more severe than others, and
Or why I guess it's more of levels of exposure, why one person can smell it but they can't eat it, but then someone else can't smell it.
The person who can't smell it also can't eat it, right? That's what you did.
Yeah. So
But I have no idea why the smelling. I don't really know how
So
it works, but I would assume it's the levels of IgE.
I think IgE is they're random combinations. So I would say probably when someone has a more severe reaction, it's more that the antibody that happened to proliferate happens to have, in the more severe cases, a greater affinity for the proteins than someone else. It's kind of it's a random chance that you find one that connects. So probably when you have a more severe reaction, you have a higher affinity receptor, which gives rise to a greater response.
But it's not
So-
necessarily the reaction that it's not the reaction that I want to know, like the exposure.
Oh, like why someone is
Why?
not as
Yeah, so.
Yeah, like you can. Yeah, you
So-
can. You, okay.
So we're gonna wrap it up in a minute. So the easy answer is yes. It's all of those things. It's what part of the peanut they're reacting to, how high their affinity is of the IgE, how much of the IgE for the peanut they have, and then the person's particular genes in terms of how much histamine do they make. You're a very sensitive person, might have many more basophils, lots more histamine, just have a lot more capacity to respond. So I think what you're hinting at, what you're getting at is it's the route of exposure as well as the amount. So the person who just smells a peanut, it's just a tiny, 1 or 2 molecules of the peanut that they're sensitive to, whereas somebody where it's just eating, they either just have those IgE loaded cells in their gut or they have to get a whole lot of peanut to activate them.
I think of the people with asthma, they don't necessarily have a lot of IgE in their blood, but it's all being produced in their nasal mucus.
Yeah, so why don't you guys finish up in the next 5 minutes? I don't know if you have anything else you want.
I guess my question is, pollen or dust is foreign as well as pathogens. So how does our body differentiate between what's harmful and what's not harmful?
I think it's just what's not self and what's self.
Well, the pollen's not.
It's specific to the
self.
proteins themselves.
well, sometimes we do react to pollen. Well, sometimes it can cause you allergy, and sometimes not. It might give rise to like IgE, IgM, which in that case it's not like actually like that severe. Like our body will deal with it, get rid of it, pretty quick, and we won't really see a reaction because it's not an IgE mediated response, and we're not getting this like crazy histamine release. Okay, or no, you guys can do it.
I just had a question about, they said in general that all of the allergies are on the rise. And has anyone looked at if it's because they're just diagnosing them more? Like my cousin, who's 20 years older than us, had a very severe egg allergy, and they didn't diagnose it until she was 20.
There's also a lot more
But now it's all of a sudden-
self-diagnosis. Within the article, I don't know if you guys picked up on it, but parents, they will say they will expose, so for example, they'll expose their children to peanuts, and they'll see some type of reaction, whether it can actually be categorized as an allergic reaction or not just any type of reaction. They're like, okay, then I'm not exposing my kids anymore because they're like self-diagnosing they're diagnosing their kid. So then they filled out this this survey saying that the kid had allergies, but that's them diagnosed. That's them saying the kid had. They never actually took their kid to a doctor, got an allergen test, and said, okay, yes, your kid positively expresses an antibody toward this antigen. So I think that definitely is a part to it. And I think it's also with, I guess, our generation and just being a little bit sensitive towards like, oh, I don't want this to harm my kid or harm myself, and kind of just like at any drop of a dime or anything that seems like a symptom of something, diagnosing it as it is without follow-up and preliminary tests.
You can also grow out of allergies.
So, right. So you bring up a very good point that your interpretation depends on what kind of study it was. This was a survey study, so it's self-reporting. There are studies that just read hospital records and take the doctor's diagnosis of allergy, and then there's people who go around the world and just draw blood and say how much peanut-specific IgE. So that would obviously be the most objective measurement of different rates of allergy in different parts of the world.
This study, I believe I found the original article. It was done for like 5,300 households in like Canada and the U.S. and it was all done through records, through medical records. And the medical records apparently did skin prick tests, which is like test for allergy.
Could you explain a little bit how you can just grow out of an allergy?
That I don't exactly know.
For 7 years, that's as
it's-
much as we know.
Yeah.
It's
It's
pretty
pretty
much
desensitization.
They can also grow into an allergy, so.
Well,
It's basically tolerance. So there's obviously positive and negative feedback, and so if your negative feedback can effectively take over and suppress those responses.
All the chemicals today.
Let's,
Imagine.
I'm sorry, you guys can keep having the discussion. Let's take a 5-minute break and then we'll get started at the end. Sorry to cut you off.